Alright, welcome back everyone. We're going to jump back into enzymes, specifically the mechanism of enzyme action today. So this lecture is going to just pertain to the function of enzymes, how an enzyme mechanically / chemically reduces the activation energy for reaction, accelerating the rate of reaction. And then the next lecture, which will be a shorter lecture, will pertain to the regulation of enzymes. So, what are we going to focus on in this lecture? Well, we're going to address what are the magnitudes of enzyme-induced rate accelerations, which we touched on a little bit in the last one. What role does transition state stabilization in enzyme catalysis? We're going to spend quite a bit of time here. How does this destabilization of the enzyme-substrate complex affect enzyme catalysis? How tightly do transition state analogs bind to the active site? Very important details in that particular part of this chapter. What are the mechanisms of catalysis? There's a handful that we're going to be paying attention to. And what can we learn from typical enzyme mechanisms? So instead of plural here, we're going to focus on a single enzyme mechanism just to get a sense of how acid-base catalysis functions specifically. So, what are the magnitudes of enzyme-induced rate acceleration? So, what we addressed in the last lecture, enzymes are powerful catalysts that can dramatically increase the rate of reaction under biological conditions. And these rates of acceleration can be anywhere from the range of 10 to the 7th to 10 to the 15th power. So that's an extraordinary change. And remember, it doesn't change the equilibrium for a reaction. Equilibrium isn't affected by catalysis. However, the rate of reaction to reach that equilibrium is affected. We need to remember that all of these reactions pass through transition state as part of the reaction pathway. We're going to be spending a lot of time dealing with that transition state. Or not a lot of time, but we're going to pay special attention to it. And the active site of the enzyme binds the transition state that's key to the reaction more tightly than the substrate. I mentioned this in the last lecture, but we're going to really dive into it here. It's an essential feature of the function of enzymes that they have a higher affinity for the transition state of the enzyme-substrate complex than it does for the substrate itself. So, moving along. So previously, let me grab a color here. Previously, we've really focused on scenario A here, where we have our free energy level on the y-axis as the reaction progresses on the x-axis, and we start with the substrate. We have this high energetic barrier transition state, delta G, and we have a product, in this case, which is favorable because it has a lower free energy than the substrate or initial reactant has. So this transition state X is our energetic barrier. Now, what I've done previously to kind of approximate the difference when showing the function of an enzyme is that we said, well, we start with the substrate, and then we dramatically reduce the energetic barrier. And even though our substrate and product have the same energy level, I don't know why that just erased itself. Let me try that again. I think I pushed the eraser button on my pen. What the main change is the significant reduction in delta G. So this is what we've looked at previously as an explanation. Now, it happens to be a little bit more complex than that, because as you can see over on B, there's actually more steps associated with this. The rate acceleration by an enzyme means that the energetic barrier between the enzyme-substrate complex and the enzyme complex to the substrate in its transition state need to be smaller than the barrier between the substrate and the transition state. So let's break that down a little bit. What we first start with is our enzyme and substrate. When those associate, there's a tiny barrier to association, but it's not a significant barrier. And following that, you get an enzyme-substrate complex. The enzyme-substrate complex inherently has to be lower energy than the sum of the enzyme plus the substrate. Otherwise, it won't happen spontaneously. It won't be favored. We simultaneously have, once the enzyme-substrate complex forms, the transition to the enzyme transition state complex, EX. This is still our high energy transition state, our energetic barrier to the reaction. However, if you look at the delta G between the enzyme substrate complex and the enzyme transition state complex, it's going to be significantly lower than a non-catalyzed delta between the substrate and transition state. And then we're still going to see the sum of enzyme plus product have the same delta from the enzyme plus substrate as the substrate to product would in scenario A. So although we approximated it in a little bit more simple terms, there's more going on in terms of the steps. Just to recap, the enzyme and substrate form an enzyme-substrate complex, which is slightly stabilized, but not overly stabilized. That enzyme-substrate complex then goes through an higher energy enzyme transition state complex, and once going through that transition state, the product is formed and separates from the enzyme, and we have separate enzyme and product. So what role does this transition state stabilization play in catalysis? First off, the binding of the substrate to the enzyme must be favorable. That's what we said first. So in this process of going, and it's reversible, enzyme to substrate plus enzyme substrate complex, step A here, call this B, call this C. In step A, which is reversible, this has to be favorable, meaning the substrate enzyme complex needs to be slightly lower energy than the sum of the enzyme plus the substrate. But it can't be too favorable. If it's too favorable, let's say I'm going to alter this a little bit. What happens if it's so favorable that we have this deep well here? Well, what that would do is create a significant energetic barrier between ES and EX. And that's no bueno, because then we wouldn't have a good rate of reaction. If you overstabilize the enzyme substrate complex, it's going to not have significant chemical motivation to leave that stable position. So it's a fine line of being more stable enough to form, but not so stable that it's overstabilized, which creates a barrier to forming the transition state. So one of the things we're going to get into, and I'm just going to keep mentioning it over and over because it's important, is that the enzyme has to have a higher binding affinity for the transition state than it does for the substrate. It needs to associate with the substrate adequately to form, but then it has to have a preference for that transition state so it can really force that to happen specifically. I think that addresses this slide. And here we go. So how do transition state analogs bind to the active site? When we're talking about transition states, we can't experimentally form a transition state in the laboratory and then probe its binding constants, because they're high enough energy where they only exist transiently. They're going to go one of two directions. They're going to go back reversibly to where they started, or they're going to go forward to a more stable product. But what we can do is synthesize structural analogs to transition states, which simulate them three-dimensionally in terms of their relative chemistries that are interesting, and we can observe how they bind the enzymes' active sites versus the substrate of that transition state which we're trying to model. So, right off the bat, let's get to the good stuff here. The affinity of an enzyme for transition states is anywhere between 10 to the negative 20 and 10 to the negative 26 molarity. This is very, very, very tight binding. So this is a really nice example of it at the bottom. Let's see here. So in the reaction of L-proline to D-proline, you go through a transition state where it becomes planar. We lose the proton, and you lose that chiral center, and you get a planar group with a charge on the center carbon here. And that's our high energy transition state. So this is not a stable compound. We have a negative charge on our carbon, and we know carbanions are high energy, and then we also have a pair of charges near each other, which are of the same charge, and that is also not a highly favored position. So this is a high energy compound. Now, the substrate, L-proline, clearly binds the enzyme strong enough to form the transition state, but then D-proline is released by the enzyme. So how strong are we looking at here? So let's see here. So we have two different analogs. We have this pyrrole 2 carboxylate and delta 1 pyrrole 2 pyrroline 2 carboxylate, and both of these are structural analogs to the planar transition state, and both of them have significantly higher binding affinity for the active site of the enzyme than the L-proline. So this is just a demonstration of how structural analogs can function as superior competitive compounds to substrates. All right, I'm back. I've just gone for about an hour and a half, but something came up. So if there's any lack of continuity in this thread on this, that's the cause of it. Just to go back through really quickly, just wanted to point out that the transition states is very high energy, so we can't test it, but we know from how well the analogs to transition states bind enzymes compared to the substrate alone, that is a preferred binding. It's much stronger. Let's see if I can advance this. And here's another. Oh, I have a redundant slide. Excuse me. Moving on. Okay, so here's another example of transition state binding and analogs and how much more strongly they bind compared to the substrate. The first case here with the yeast aldolase reaction, A, is looking at the initial substrate going through an indiolate transition to the fructose 1,6-bisphosphate final product via glyceraldehyde 3-phosphate reaction. That's the enzyme. And so I just put product in here. And you can see that through coordination to zinc in the indiolate transition state, that's a very high energy compound. We have some charges. They're slightly stabilized through pairing with the zinc here, but this is a high energy compound which rearranges significantly. Now, it turns out that this particular analog, phosphoglycolohydroxamate, is got significantly higher binding to the enzyme than the substrate does. So this is a good example. And over here on the other side we have this deaminase reaction, and you can see the same idea where the analog to the transition state is actually a preferred compound to bind the enzyme at the active site versus the substrate, in this case adenosine. So everyone here has been through organic chemistry, but it's worth revisiting just a little bit the rationale for pushing electrons. When we are pushing electrons, when you move a pair of electrons, two electrons, it's always denoted with a double-sided arrow. And in the case of a radical reaction where we push a single electron, that's denoted with a one-sided arrow. So next semester, when we're doing a little bit of work out the gate with mechanisms, a lot of mechanisms in enzymes actually occur through radicals. And we'll actually have to differentiate quite a bit with the double-sided arrow versus single-sided arrow to rationalize the chemistry that's taking place. Now, taking it back to the two-sided arrow exclusively, if you're breaking a bond, like in this scenario here, call this scenario A, B, C. In scenario A, if you break a bond between A and B, and you move both electrons that are present in that bond to, in this case, let's say B, B is going to have an excess of electrical charge, assuming it was neutral, like it is listed here, and that extra electrical charge is going to result in negative charge. We could actually probably even put a, well, we can't necessarily put a lone pair, but if it's a base, we can say lone pair base. Breaking that bond and moving the electrons away from A results in a loss of electrical density, which results in a positive charge. Likewise, if we're forming a bond between a base, B, lone pair, minus, attacking a electrophile A, so we have just basic nucleophilic attack, we can form a bond by tracking a pair of electrons that now become shared between two nuclei, and this results in satisfying both the negative charge of B, positive charge of A, and then we end up with a neutral species. Why we're paying attention to this so much is because of proton transfer. When we're doing proton transfer, that's not radical based. Proton transfers revolve around pairs of electrons moving. And we're going to pay extra attention to it because it's been estimated that about 75% of the steps in enzyme reaction mechanisms involve proton transfer. They come on, they go off, they come on, they go off. And at the end of this lecture, you'll see some good examples of repeated proton transfers that facilitate different chemistries. So, one of the odd conventions of biochemistry is that if a proton is donated or accepted by a group on an enzyme, it's traditional to represent that group as B for base, even if it's protonated and behaving as an acid. And that's pretty odd. But a lot of these groups can be acidic and then be basic because of the conjugate nature of acids and bases. So if it is a target for an acid-base reaction, it's really common to list it as B. And that's what we're seeing here in C, is that here we have a nitrogen with a lone pair acting as a nucleophile, attacking this proton, breaking the bond between the hydrogen and the base, and pushing the electrons to the base. Now, it should really have a lone pair here. It's acting as a base. Now, it's important because this is acting as an acid. It's donating a proton. And the nitrogen's acting as a base. But on the other side of the coin, now we have a base acting as a base, and what originally acted as a base is now a conjugate acid. So, biochemists are not necessarily the most comfortable with organic chemistry, and vice versa, and there's some differences in convention in how they display these chemistries, and this just happens to be one of the conventions you have to get comfortable with, even though perhaps it's not as elegant as it could be, but it is the convention. So, I have more redundant slides. Moving on. So, this is a nice example elaborating on mechanisms and the convention of the base. When we're doing proton transfer specifically, it's really important to appreciate that a nucleophile will change into an electrophile and vice versa. The base can become an acid and the acid can become a base. And particularly when we're dealing with histidine, like we are in this example. And it's just the labile nature of proton transfer where a base, starting here, can act as a base, take the proton, breaking the bond to satisfy the positive charge on the nitrogen. Then moving on, we now have a nitrogen on the histidine, which is able to act as a base instead of an acid like in the previous step, can reach out, grab a proton, breaking a bond, transferring electrons to our oxygen and charging that oxygen. That puts the proton back on the nitrogen and now can act as an acid again. So this is the nature of how enzymes take protons and give them away, and take a proton and give them away as part of a chemical reaction, facilitating it, versus the mechanism you're probably most comfortable with, where you just have an aqueous environment and with a little bit of an acid or basic catalyst can take and reclaim protons with water. Here, because it's not just floating around an aqueous solution, we have to have other mechanisms to accomplish it. And as we'll see over and over and over moving forward for the rest of this and the next semester, histidine typically plays a really important role in this kind of proton transfer. So pay particular good attention to the presence of histidines in binding pockets. Now, water can act as an acid or base. Even though we're not just taking place in aqueous solutions, it's not uncommon for it to be in the binding pocket and participate as part of the event. So in this particular case that we're looking at, you can see the functional group of a histidine in its basic form doing an attack. And this is a little bit of a simplification. What should really be shown is we have the nitrogen doing an attack on the hydrogen, breaking the bond. This particular bond then transfers electrons to the oxygen, forming a hydroxyl group, which can do an attack. So it's a little bit of a simplification as drawn. Then that nucleophilic attack pushes electrons to the oxygen, which can be stabilized a variety of ways, but like you would expect when you have 3 lone pairs around the oxygen, one of the fates is to have a lone pair come back down and kick off the group that attacked, or kick off a good leaving group, or reach out for some subsequent chemistry, or be stabilized by a metal cation. So getting a little bit ahead of ourselves there, but water can participate in acid-base chemistry. It's just not to the same extent, and it's more facilitated than it is just floating around in aqueous solutions. So what are our mechanisms of catalysis? There's 6 we're really going to focus on. We're going to focus on the enzyme's facilitation of the formation of what's called a near-attack complex, or the NAC. The near-attack complex, well, we'll get into it in more detail in subsequent slides here. We're going to focus on how protein motion is essential for catalysis. We don't want an overly stabilized protein that is incapable of fluid motion. We're going to talk about covalent catalysis, general acid-base or acid-base catalysis, low barrier hydrogen bonding, and metal ion catalysis. So these are all functions that we're going to have to pay attention to, or mechanisms in which an enzyme can facilitate a chemical reaction. So let's talk about the near-attack complex first. We have both X-ray crystallographic data and computer modeling data that indicate that reacting atoms and nuclei in catalytic groups are very precisely oriented. And that very precise orientation is critical for the function of the enzyme to perform that particular chemical reaction efficiently by reducing the activation energy. So the reason for this is because the reacting atoms get into van der Waals distance, or actual orientation, where their orbitals both are proximally oriented, they're close, and they're also positioned at the right angles to facilitate the transfer of electrons. Now, one of the reasons that reactions require energy is because of all the random motions between two things. So when they collide, they have the right energy, but also the right orientation. Things can bounce off of each other if they don't have the right orientation, but they have the right energy. And we can see that that appropriate orientation sometimes can only occur a tiny, tiny fraction of a percent of the time under normal conditions. But when an enzyme facilitates the orientation of different compounds, that you can get that appropriate orientation anywhere between 1 and 70% of the time. And that's a really important distinction. So a good example of this would be, say, the SN2, basic nucleophilic reaction for an SN2 reaction, where you have a nucleophile attack specifically from the back. And that specific attack from the back of the compound is essential because this occupied orbital on the nucleophile needs a place to put its electrons. And as we're forming this transition state, forming a new bond between the nucleophile and breaking the bond to the leaving group, we've overcomplexed to carbon, but we don't have full sharing of electrons to two of these bonds. And these electrons are moving from the nucleophile into a vacant antibonding orbital behind the carbon here, which is oriented because of the bonding orbital between the carbon and leaving group. So it's that very, very specific orientation which is required. And what the enzyme does is, instead of making these two things just happen to bump into each other at the right angle with the right velocity, is it takes them and just physically puts them there. It aligns the occupied bonding orbitals or lone pairs with the appropriate antibonding orbitals so that everything lines up beautifully, dramatically reducing the activation energy, because now everything is where it's supposed to be to facilitate the transfer of electrons. And that's what a near attack conformation is. And this is another example of near attack where you can see you not only need to have the right distance between nuclei, but you also need to have the right angle, plus or minus a couple degrees, to really facilitate this nucleophilic attack. So in this case we have a carboxylate which is attacking a ketone, and everything just has to be lined up right to facilitate this type of reaction. Oh, that's not a ketone, that's another carboxylate, or an ester or ether. Now, if you look over to B, to the right here, it's our favorite way to look at reactions, which is our free energy on the y-axis and reaction progress on the x-axis. And what's really important to look at here is that at the active site of an enzyme, the NAC forms more readily than in the uncatalyzed reaction. So in the uncatalyzed reaction, you have a much higher energy to reach NAC, because of the probability associated, thermodynamic probability of getting it there, versus it being facilitated via coordination with the enzyme. So it's just another way to demonstrate that the enzyme is facilitating the formation of the near attack complex. And this is just a pretty picture, which is looking at the alcohol dehydrogenase enzyme in liver, which is everyone's favorite enzyme after finals week because it well facilitates the breakdown of ethanol after it's been consumed. And this is just the near attack conformation, kind of demonstrating, you know, a nice picture of how perfectly everything has to be aligned to accomplish the chemistries that take place in biological systems. So, the next point that's essential to make in this lecture is that protein motions are essential for catalysis. Proteins are just moderately stable, remember. They're stable enough, but they're labile enough where they can move. And that's really important because that motion is absolutely critical. When the substrate binds the enzyme, that binding event causes a conformational change. And now that they're connected, the conformational change of the mass of the enzyme helps drive twisting the substrate and moving it and lengthening bonds and changing bond angles to what would be non-ideal if it weren't complexed to this other larger structure. But the new preferred state of the enzyme substrate complex, even though it's in lower energy, can take that substrate into a higher energy configuration. And that's the transition state that's modeled. It actually forces things, and lengthens things, and reorients things so that they're just perfectly aligned to do the reaction that we want to facilitate biologically. It's just a spectacularly elegant system. So these motions are essential for substrate binding. They're essential for bringing the catalytic group into position, for the formation of the near attack complex, for assisting in the lengthening and shortening of bonds, for making bonds and breaking bonds, and ultimately converting the substrate into the product. And then once the product's formed, it's released because of the lack of specificity for the binding site, and the enzyme returns to its native conformation, ready to accept another substrate. You can see in this graph that some residues in the active site are highly mobile over the course of it binding and releasing a substrate/product. And this is just to give everyone an idea that yes, there's an enormous amount of mobility in that active site. There's a lot of chemistry taking place, and some of the residues are absolutely essential to be very mobile. So another mechanism by which enzymes function is through covalent catalysis. Very commonly, you'll get a scenario where a nucleophile will attack a residue on the enzyme, cause a covalent bond, and then you'll have a subsequent covalent reaction which causes the substrate to become the product and become released. Or this can happen through a series of covalent interactions. And these are a couple different examples of that effect. Let me number these. Let's go A, B, C. And these are just examples of this where you have an enzyme with a basic group, in this case denoted X, that's committing a nucleophilic attack to a phosphate. The electrons are originating from the base, attacking the phosphate, pushing the electrons up to the oxygen. This causes a charge to form, and we also have more bonds to the phosphate, so this facilitates a leaving group leaving. And the electrons come traveling down, reforming the double bond, and this causes the R group, leaving group, to be kicked off. Let's see, another good example, and this is actually, B is almost identical, except that instead of having this acting as a leaving group, instead we have a generic group Y acting as a leaving group. So very similar chemistries. Now, acid-base catalysis is any catalysis in which a proton is transferred in the transition state. And this can happen through specific or general mechanisms. Specific acid-base catalysis involves the donation of a proton or hydroxide that diffuses passively into a catalytic center, whereas general acid-base catalysis involves acids and bases that are not just free protons and hydroxides. This is a really good example of catalysis of para-nitrophenylacetate hydrolysis, and it can occur either through general or specific mechanisms. First off, through the specific mechanism, let me grab here, we have hydroxyl, and it's got those 3 lone pairs around the oxygen that can facilitate the nucleophilic attack on the carbon, pushing the electrons up to the oxygen, which is leaving us with our charge here. And this is the same high energy intermediate that will then result in the loss of the leaving group when the electrons push back down and kick that off. So this can happen through the specific mechanism with the free hydroxide, or it can be facilitated with the residue on a histidine, which we're seeing here. Basic nitrogen attacks the proton, pushes these electrons to the oxygen, charging it. We now have hydroxyl, which can do an attack, and it's just facilitated, more facilitated mechanism. This is a really neat phenomenon. We were talking about the near attack complex, how everything gets aligned perfectly. Well, one of the great examples of this is the low barrier hydrogen bond. So remember, hydrogen bonds are always thought of as like 10% covalent bonds. They're longer, substantially weaker than classic covalent bonds, but they have some covalent character, which means there is a degree of electrical sharing through that distance. Now, why that's important is because when you observe hydrogen bonds in nature, they have that longer bond distance because it's not being facilitated to be shorter. In enzymes, you can take a hydrogen bond and through physical manipulation of the bond distances, push 2 nuclei closer together. And if they're already hydrogen bonding, that physical pushing of them together gets the orbitals closer to one another, closer to that van der Waals distance. And you can see the barrier for the physical transfer of the electron between the nuclei, which was large, becomes shorter, and at some point becomes negligible, and you can easily transfer the proton from one nuclei to another without there being an energetic barrier to it, because the nuclei are all aligned spatially in a way that facilitates it. So there's a huge advantage to the transfer of protons enzymatically because you can facilitate it via low barrier hydrogen bonding. Another mechanism, in addition to the low barrier hydrogen bonding, is even more elegant and used quite a bit in enzymes and in biological chemistry, is quantum tunneling. And everyone's like, don't worry, you don't have to have physical chemistry to understand this. It's just quantum tunneling. It's not rocket science. Sorry, bad joke. What this is tunneling provides a path around the normal energies of activation, because according to quantum theory, there's always a finite probability in a particle can appear at another side of an activation barrier in a reaction step. And that's what we're seeing here, is the concept that a particle can move across an activation barrier in the same sense that an electron can move between orbitals without actually passing physically through space. It is in one orbital, then it's in another orbital, but it doesn't pass between them physically. And what dictates this is the unique fact that matter has wave character, as shown by the de Broglie equation. Now, for tunneling to have an effect, to play a role in a reaction, the wavelength, I should point to it here so everyone can focus on it. The wavelength of the particle that's being transferred needs to be similar to the physical distance over which it's being transferred so that it can act as a wave to hop through space. And that limits the type of particle that can behave in quantum or participate in quantum tunneling to protons and electrons based on those physical distances of their wave character. So that's good because we can participate in proton transfers as well as electrical transfers. And if you don't have to, if you get everything the right distance, there's a high likelihood that you can get a quantum tunneling effect which bypasses energetic barriers. Additionally, we need to discuss the effects of metal ions in catalysis. One of the things I've done a number of times, I would do a nucleophilic attack, which involves in a second nucleophilic attack. And let me grab a color. So we have our nucleophilic attack. I really wish we had our lone pairs drawn in here because it really shows the nucleophile anisomer. This hydrogen leaves. Those electrons go to the oxygen, creating a hydroxide. That hydroxide then does a nucleophilic attack on the carbon, pushing the electrons, because we've exceeded our octet, up to the oxygen because it can house them due to its electronegativity. And typically, once up on the oxygen, they can do one of three things. They can come back down and kick off the group that just attacked, or they can kick off another group, or they can reach out for another chemistry. In this particular case, the presence of a zinc, or it could be any other metal that's cationic, that's perfectly located next to this reaction, carrying a positive charge, is able to relieve that extra electrical density from the oxygen. So it stabilizes that intermediate, allowing more time for something else to take place. The other way to view this is that the zinc, because it wants electrons, is pulling electrons from the oxygen, which makes the carbon better nucleophile, which facilitates that initial attack. So there's really a chicken and egg argument there, but both rationale apply to how metal ions are essential in a lot of catalytic events in enzymes. And what I want to do to wrap up this lecture, since we just hit you with a whole ton of content, is just provide one detailed mechanism. And I don't want everyone to know this one by heart, even though you should know what the catalytic triad is generally, because that is important. It's a classic. Let's talk about serine proteases as a good first example, or in this case, our prime example, detailing some of the ways by which an enzyme can function and how detailed it can be and how elegant it is. So the serine proteases are a pretty darn good example of acid-base catalysis, as well as covalent formation and breaking of bonds, and how substrates are passed throughout the binding pocket and reoriented to do a series of chemical events. So serine proteases include trypsin, chymotrypsin, elastase, thrombin, etc. All these involve proteolytic cleavage of different specific residues in amino acid chains, are essential for a lot of our biological chemistries, as well as manipulations of peptides in laboratory settings and doing digests. So they're called, in this case, serine protease because serine is part of the catalytic triad of serine, histidine, and aspartate, or aspartic acid, and all of these are homologs, but there's some differences between them that give them different specificities into how they attack and what they attack. But the triad is typically agreed at as being His 57, asp-102, and serine-195. And this is just a diagram of how these R groups are oriented to one another so that they're always being coordinated, where the aspartic acid or aspartate is coordinated to this proton so that it can exchange back and forth, and how the lone pair on this nitrogen is coordinated to the hydroxyl on the serine so that that hydrogen can move back and forth between the oxygen and the nitrogen as part of the protons shuffle that's going to be taking place. Okay, this is a little bit redundant, so you can peruse this, but I'm just going to hop right into the mechanism itself. So this is a 2-slide mechanism with 9 steps. So, like I said, you don't have to know this one by heart. This is just for demonstration purposes. Note that we've got this binding pocket, and we've got our triad oriented. And as the substrate comes in, what happens is a series of binding of the substrate, which results in the formation of a enzyme substrate complex. And what takes place is, remember, our oxygen is coordinated to our hydrogen and our nitrogen is coordinated to the proton on the serine. And to get this whole party started, we need to make a nucleophile. So the histidine grabs the proton off of the serine. This puts those electrons that were between the hydrogen and the oxygen on the oxygen. This is a very strong base. And now it can act as a nucleophile, attack our carbonyl, pushing the electrons up to the oxygen. Now, once we have this new complex, we can get an attack back where now, once it's a very strong nucleophile, now it's bound to the carbonyl, and we have a new nucleophilic attack take place between the substrate, and it's going to grab the proton right off of that histidine, putting the electrons back on the nitrogen. This is going to result in a internal chemical reaction on the substrate, where the electrons that were being housed up on top of the carbonyl, or what was originally a carbonyl, are going to come back down to reform the double bond. That's going to break the bond between the carbon and nitrogen, causing a leaving group to fall off. This is a bond cleavage event, which is why it's a protease. And then as it leaves, we get the release of the amino acid product. And then we have to have a series of resets and something that kicks everything back out of the pocket. So the histidine residue with the nitrogen is able to grab a new proton off of water. This forms a hydroxide, which can do a nucleophilic attack on carbonyl that's been reformed, pushing electrons back up. Then they push back down, re-grab a proton, kick off our histidine, and minus just loss of the proton off of the carboxylate, reforms product, which then leaves the group, and our triad is reset so it can do it all over again. If you think about all the little tweaks and alignments that take place during the sequence of events, imagine in the binding pocket all of the little manipulations, all the subtle changes to spatial orientations, the lengthening, shortening of bonds, alignment of nuclei, and the speed at which it takes place. It's just astonishing. And this is just a really elegant example of proton transfers. That's it for this lecture on mechanisms. We're going to have another lecture, also briefer than this one. I'm guessing about 35-40 minutes on regulation. And then we'll have 2 more lectures after that, a little quick summary on carbohydrates, lipids, and membranes, and that's going to be the semester. So I hope everyone's enjoying this. I think it's an amazing topic, and I'll be talking to you guys in a couple more days.